So today what I want to talk to you about, of course, is the result of my research these last about 5 years on the food of the gods, the food of ambrosia beetles. And I'll explain a little bit more about this interesting title right away. So there we go. Ambrosia was the food of the gods in Greek mythology. It was what they ate. It gave them their immortality. It was their food alongside their nectar, which was the drink. But today, we're not talking about Greek gods, we're talking about ambrosia beetles. There's another connection here too, and I'll save that for the end. So, fungal agriculture, actually agriculture in general, has only been developed 4 times in animals. There were the ambrosia beetles about 100 million years ago, and it evolved at least 11 times inside of these beetles. In attine ants, you might know of leafcutter ants, which farm fungi. That's why they gather leaves. It's to fertilize fungal beds. It evolved only once in those ants about 50 to 67 million years ago. In the Macrotermitidae, which farm Termitomyces fungi, this has also evolved only once, a little bit younger, about 31 million years. There's one more group of animals that evolved agriculture. Can anyone think of what that might be? I'm looking for an answer.
Humans.
Yes, that's right. Great. It's us. And only one species of Homo has evolved agriculture, actually. It's only evolved once, and only about 10,000 years ago. So, today I want to talk about the first, the oldest, maybe the most speciose, and the most derived of these agricultures. Here is a beautiful picture showing some diversity of wood-dwelling weevils, or scelidids. You can see some of them stay just under the bark, and they kind of carve these intricate galleries. Some of them go deeper inside. Those bark beetles that form the intricate galleries on the surface are feeding on the inner bark. They're actually eating part of the tree matter. Ambrosia beetles don't do this. They go deeper into the wood, into the sapwood, and most of them don't feed on the wood, at least not as a primary component of their diet. Instead, they're feeding on fungi. You see, the sapwood where ambrosia beetles make their galleries is pretty nutrient poor, especially compared to the inner bark, but it's a niche they exploit using fungal symbionts. If you look along the galleries of ambrosia beetles, they are lined with a carpet, a thick mat of mycelium, of fungi that aren't just there accidentally, but are actually being cultivated by the beetles as a food source. Here's a typical ambrosia beetle gallery. You see the white ambrosia fungus growing along the walls. You see a mother here who's tending to her young, before I split this tunnel open. And if you look at this fungus more carefully, you'll see specialized structures that are not for dispersal, but for feeding. Big plump spores, big nutritious spores that are on top of conidiophores made to disarticulate and made to present themselves for grazing in the gallery. There's a certain degree of co-adaptation here. The fungi need to provide the beetles the right kind of nutrients, and the beetles need to develop the behavior to tend these fungi, take out weeds, transport them to new locations. It's the transporting to new locations that involves maybe the most amazing part of this symbiosis. So if I take this beetle here, crack it open, you'll see there is an organ here, a mycangium. And if I take that mycangium out by itself, we plate it under a microscope slide, and look closer, it is full of a pure homogeneous mass of fungal propagules. All ambrosia beetles have mycangia. They are organs used to store and transport fungal propagules of their symbiont. The fungi actively grow in here, as you can see by the spore mass here on the beetle that has actually emerged from the mycangium. And there's co-adaptation or co-adaptation here too. The mycangium needs to provide the right nutrients for the fungi to grow, and the fungi need to be able to enter a yeast state. Almost all ambrosia fungi are dimorphic. Even though they're filamentous in the gallery, they make this yeast phase for when they are growing and being carried in the mycangium. But this is only one example of a type of mycangium. There are many types of mycangia. In fact, the word mycangium just means any structure that these beetles use to carry fungi at all to new locations. On the left here, you can see some of these mycangia are a lot smaller. Where the yellow arrows are pointing, you can see smaller white cavities, oral pouches, little shelves near the elytra, small cavities under the arms. But other mycangia are larger and more complex. On the right here, you can see the white structures inside of the beetles' bodies. Those are the mycangia. these are what we focused on for this study. Specifically, the mesonodal pouch mycangia, the procoxal coil mycangia, prothoracic pleural, and pronodal discs. I'll be using these color codes for the rest of the talk and these same silhouettes of these mycangium types so you can keep them straight in your head. Ambrosia beetle symbiosis evolved multiple times. I mentioned this at the beginning, at least 11 times. Each of these evolutions was from bark beetles, and in each case, the ambrosia beetles began the symbiosis thanks to a mycangium. So a lot of these mycangia here in yellow are the smaller type. Again, we are interested in the 4 lineages with these really large, complex mycangia. Most known ambrosia fungus symbionts, cultivars, are in the genus Raphaelia in the order Ophiostomatales. But interestingly, these 4 groups of ambrosia beetles with the particularly large mycangia had all been implicated with different symbionts in the order Microscales. They'd usually been called Ambrosiella, but this was a genus with not a lot of data to back it up molecularly and a relatively small amount of studies on it. So you can see here, each of the 4 types were associated with this Ambrosiella. Only 2 of them had been confirmed molecularly. Some of them were only known from 1 or 2 studies, and they were illustrated, and that's about it. This is what Ambrosiella looked like in 2014, just before I began this study. You can already see that it's not a monophyletic group. There's the non-ambrosial fungus Ceratocystis adiposa here between the Ambrosiella taxa. So we already knew it wasn't a monophyletic group of symbionts. And the 2 groups happen to correspond with these 2 types of mycangia, the mesonodal in green and the pleural in orange, but we weren't sure what the symbionts of the other 2 groups were. The goal of this study was to collect beetle species representatives from each of these mycangium types, isolate their fungi, characterize them, molecularly identify them, and see if this trend held beyond the limited studies that were available previously. Generally, we collected beetles using flight traps, such as the Lindgren trap, which uses ethanol to lure the beetles in. They hit this large black column and they fall into a collecting cup. You can also just cut the galleries directly out of wood and remove the beetles yourself. We actually did very little of this ourselves. We really relied on collaborators around the world to do this for us and send the beetles or galleries to us. I want to impress upon you now how small these beetles really are. Here is the largest and smallest ambrosia beetle species in the United States, and here's a dime. So thankfully, it was not me having to pull these very, very tiny beetles out of wood and out of the air all the time. We had collaborators across the United States willing to send us this material and around the world, which helped us get a relatively comprehensive sample of each of these 4 mycangium types from a decent spread of geography. Typically, especially at first, we ground the beetles into slurries, dilution plated them out to get quantification from the mycangia, and isolated and characterized any different morphotypes that appeared on the plates. But eventually we found it was just as effective to just crush a beetle and plate it directly after surface sterilizing to get rid of contaminants, and just look for what grows out of the mycangium, or to take from mycangial spore masses on the outside of the beetle directly, or to just use sterile tools to isolate from the gallery growth itself. For DNA sequencing, for barcoding purposes, we use the standard fungal primers, ITS1F and ITS4, to amplify the internal transcribe spacer region of the ribosomal DNA. Many of you will be familiar with this in fungal barcoding. And for multi-gene analysis, we used a concatenated set of 6 different genes, and we used those to do Bayesian analysis with Mr. Bayes, maximum likelihood with RAXML, and we also did some divergent state estimates, which I'll show at the end, using Beast and Beauty. So we managed to get several representative species from each of these mycangium types, 9 species with the mesonodal mycangium, 9 with the procoxal coil, and 7 with the prothoracic pleural. In bold here, I have the species where we were able to successfully isolate a living ambrosia fungus culture, which allows a lot more room for morphological study and allows you to get more genes. The ones not in bold, we only were able to sequence DNA from mycangia or galleries. So for the mesonodal mycangia, we got representatives here of 4 different genera. All these genera come from a single origin of this mycangium. They're a monophyletic group. We collected them here in Ames, and we got them from Asia, Europe, etc. The mycangium looks like this. It's a pouch just ahead of the scutellum of the beetle, invaginated into the back of the pronotum. If you remove the mycangium in the surrounding tissue, it looks like this. The arrow there points to the fungal mass inside of the mycangium, and it's filled, of course, with pure yeast-like propagules. All the mesonodal mycangium symbionts yielded DNA sequences that place them along with the type species of Ambrosiella, Ambrosiella xylobori. So these were legitimately Ambrosiella species. This wasn't surprising. The mesonodal mycangium was the most well-studied mycangium before this point, but at least we confirmed that. And we were able to report a few new associations of fungi with unstudied beetles, which led to the description of 3 new species here. Now, you might notice within these species that it seems pretty species specific. For example, Xylosandrus germanus from across the United States yielded the same exact ambrosia fungus species as Xylosandrus germanus from Europe and from Korea. And at first we did think they were species specific. This has turned out to not be quite as true, and I'll come back to this in a moment. Ambrosiella looks like this. It makes these beautiful pigmented cultures with little droplets of pigment, and they smell like bananas. The cnidophores look like this. They are these large globose cnidia on the end of towers, and when they detach, they give this characteristic look of a single cell with just the penultimate cell of that cnidophore attached to it, and sometimes they look like little mushrooms. It's pretty cool. So like I mentioned, this was not species specific. A study by Lin et al. In 2017 showed that some of these species can actually be found from several different ambrosia beetle species with mesonodal mycangia. Beaveri was the most diverse with, I think by now there's 8 different species associated with it. Our data since then has confirmed this. Probably our data was skewed by looking at introduced populations in the United States and Europe rather than their native ranges, but we'll see. Regardless, 18 of the 18 studied mesonodal mycangium species have yielded Ambrosiella symbionts. So this seems like a pretty clear relationship between a genus of fungi and a type of mycangium. Quick side story, while we were studying the ambrosia beetle Anisandrus maisch in Ohio, or from Ohio, which also has a mesonodal mycangium, we noticed these little structures growing in the galleries, these little spherical bodies filled with what looked like ascospores. We had seen immature bodies like this in other cultures of Ambrosiella, but weren't quite sure what to make of them. You see, ambrosia fungi have always been assumed to be strictly asexual clonal lineages, so it was a little surprising to suddenly be seeing what appeared to be Cleistothecia, sexual fruiting bodies. Now, close relatives in the Ceratocystinaceae do make sexual bodies, Parathecia, with spherical bases and these long necks where they produce sticky ascospore drops to stick to the outside of insects for dispersal. But ambrosia fungi don't need this. They're carried in mycangia, and it was generally thought that because they're propagated in this yeast-like form, they wouldn't have the opportunity to do effective sexual recombination. But sure enough, on studying these structures further, not only were they full of ascospores in the gallery, but isolations of this Ambrosiella species also produced fertile ascocarps. So we were able to report for the first time a fertile sexual state in a mycangial ambrosia fungus. Don't know what the purpose is here, if it's an alternative food source or dispersal, but we reported as such in a paper in botany last year. Also, in studying Xylosandrus compactus, the ambrosia beetle in Mississippi, which carries the type species of Ambrosiella, we once again found these structures. Since then, we found immature structures in 4 Ambrosiella species, fertile structures in 2, and a French botanist, Bovary, in 1910 also illustrated them, but that went unnoticed for a while, so he helped me out. So it seems pretty likely that Ambrosiella produces sexual recombination in nature, maybe cryptically. I will note that the species we got in culture from Anisandrus maisch appeared to be homophallic, so it could be they mostly mate with themselves. Okay, on to the next type of mycangium, the procoxal coil. You'll see we studied 9 different species here. They're all in the genus Cortholis. It's the only genus known to have this type of mycangium. It looks like this. It's a wound spiral tube inside of the beetle's body that feeds through a little opening under its armpit. Here I've dissected one out from Cortholis papillans. You can see it's this winding tube. It's really cool. And thanks to collaboration with Craig Bateman down at the University of Florida, we were able to get 3D CT scans of these organs as well and report for the first time some diversity in the morphology of these mycangia. Some of them wind quite a bit, some of them are more spiral. The fungi we isolated from these Cortholis beetles, formed a single monophyletic group that was separate from ambrosiella in green up there at the top. We were able to describe 3 new species here from these beetles. 9 of the 9 Corthless beetles all yielded either cultures of this fungus or DNA sequences of this fungus, and we gave it the new genus name Meredithiella after Meredith Blackwell, who I actually knew during my time at LSU, and it was a big inspiration for me to get into mycology. And we reported this in a paper that should be out in Mycologia this year. Meredithiella looks like this. It typically makes little side chains on its conidiophores. It's quite different from ambrosiella, although the conidia that detach can sometimes look pretty similar to ambrosiella. So we call these Allurioconidia. It's a specialized form of conidiophore, although one of the species didn't seem to produce them. Now we did find immature fruiting bodies in one of these species, but we weren't able to get it to fruit or find ascospores to see what they look like. Probably, or maybe that exists in nature though. The third type here, the prothoracic plural mycangium, we studied 7 species, all in the genus Tripodendron, the only genus currently known to have this exact type of mycangium. It looks like this. You can see I've dissected one here. It's a sac of sorts that travels upwards and then folds downwards again, and it's in the pleural cavity along the left and right side of the beetle's body. From these studied species, the 7 of them yielded species in this group here. You can't see ambrosiella here, but this is separate from ambrosiella. These are part of that group with ambrosiella ferruginea, which I showed near the beginning. It was already known to be separate from the main ambrosiella group. We described, or in the process of describing, 3 new species here from these Tripodendron beetles. 7 of the 7 Tripodendron yielded this fungus. We've resurrected the name Phyalophropsis for this group. It was available, it was named in the 1960s, but had been synonymized to ambrosiella since. We brought it back from the dead. So Phyalophropsis looks like this. It produces its conidiophores inside of deep phyalids, which means the conidia are made inside of the cells that they're produced from, and then kind of emerge, kind of like a cannonball coming out of a cannon, rather than just budding from the end of the conidiophore. In the galleries, they make palisades of spore-producing structures like this that the beetles feed on, which is quite unlike ambrosiella, and they display a surprising amount of morphological variation when they're grown in culture. So this was one of our first multi-gene trees. This is a combination of 18S and elongation factor in a Bayesian analysis. And it made it pretty clear that these 3 groups were legitimately separated from each other: Phyalophropsis, Meredithiella, and ambrosiella. So our thinking at the time was, maybe this is 3 different domestications by the beetles. Maybe this is 3 different evolutions to ambrosia symbiosis within these fungi. And we hypothesized as much in a paper in fungal biology back in 2015. And this story was great. It all made sense. It seemed like everything was fine until we were visited by another friend. So I mentioned a little while back there was a 4th color to use mycangia. It was the purple pronotal disc. I haven't forgotten about it. I just decided not to mention it until now. So the story gets a little more complicated. The first 3 mycangia I talked about are all derived lineages from other ambrosia beetles that might have had different mycangia, that might have started with different symbionts and cultivars, and then switched to these different types of mycangia when picking up their symbionts. The pronotal disc is different. It's an entire tribe of ambrosia beetles in the tribe Scelidoplatypodini, who all have this nearly identical mycangium type, which seemingly coincides with the origin of ambrosia symbiosis in that group. And it was known that African and Asian Scelidoplatypus, one of the only 2 genera in this tribe, were quite distinct lineages from one another. We were able to collect specimens from 6 of the Asian species and 3 of the African species of Scelidoplatypus from a couple of different locations for each. We were able to isolate cultures from at least 1 beetle in each of these groups, and we were able to get 1 specimen to study of the other genus in this tribe, Romansis, which is only known from Madagascar. Both of these genera, Scelidoplatypus and Romansis, have nearly identical mycangia. There's a conspicuous hole in the back of the beetle's body, almost like a blowhole, underneath which sits a reticulated, glandular, UFO-shaped body. And if we cut the beetle off like this and split it open, you'll see a cross-section that looks something like this. Near the top, there's a little onion-shaped cavity. That's the mycangium. If we zoom in, it looks like this. These are usually filled almost completely with fungal propagules during the right time of year in some sort of matrix, but they're also lined with these hairs or setae that grow from the walls and generally towards or out of that little hole, the opening of the mycangium. It's thought that these help guide the spores out, but they might also provide some support to the mycangium or other functions. The inside of the mycangium of Romances had never been studied before, so thankfully we were able to open one of them up and take a look. It was a similar morphology. It was a disc-shaped organ with hairs lining the walls, but the hairs were quite different than those of the other beetles in Scledoplatypus. In fact, African and Asian Scledoplatypus differ quite a bit in their mycangium setae. In the African, they're pretty simple. They grow directly off of the mycangium wall, and they come in two sizes. In Asian Scledoplatypus, the setae are supported on these reticulated conical structures that project into the mycangium lumen. And in Romances, what I observed was they did have these reticulated structures, these cones, but there were no setae on the top of them. The setae were instead on these cylindrical, non-reticulated columns, which don't really resemble the other setae. So, three distinctly different types of setae, corresponding with pretty divergent relationships, especially between Romances and Scledoplatypus. And even though it seemed like the African setae was the most primitive type, I mean, it's just hairs growing directly off of the wall, phylogenetic analyses have shown that Romances is apparently the basal member of this group. So, we'll see. Going back to the ambrosia symbionts I talked about before, in our nice little three groups, let's see where the Scledoplatypodini symbionts shaked out. The African Scledoplatypus symbionts were here next to Phyllophoropsis, but the Asian symbionts were related to Ambrosiella instead. And Romances had a symbiont that seemed like it should be treated right inside of Ambrosiella. It was just outside, but it was a lot more related to these Ambrosiella species compared to the Asian Scledoplatypus symbionts, and it seemed to share a mycangium morphology with them as well. So this was pretty surprising. We had expected a fourth, novel, separate lineage of fungi, like we'd already found with the other three mycangia, not interspersed symbionts throughout the diversity here. The African Scledoplatypus symbionts had really simple kinetiophores that really didn't resemble Phyllophoropsis very much. And we found fertile sexual bodies in the cultures from one of the species that produced gigantic ascospores, bigger than any known in the family. The Asian Scledoplatypus symbionts did look similar to Ambrosiella, but their allurioconidia were much larger than Ambrosiella, and they had a different culture morphology as well. So this was interesting. We had three groups of apparently derived ambrosia beetle lineages that only had single consistent symbionts across their diversity. And then we had another lineage in a tribe which has had this mycangium since the beginning, carrying three different symbionts across its diversity. And also, this pronotal disc mycangium was thought to be part of one of the oldest lineages of ambrosia beetles, certainly older than these other three. So at the time, we thought maybe this was the origin of symbiosis in this family. Maybe the Scledoplatypodini domesticated these symbionts first, and all of the other symbionts we were observing came from those through trading between beetle lineages. So we produced a 6-gene Bayesian tree here, the most comprehensive in the family so far. Also, I've got the novelty stars back. We're going to propose new genera for the Asian and the African Scledoplatypus symbionts. At the moment, that would be Wolfgangiella and Toshionella, which I'll be using for the rest of the talk. Those are not published yet. And we were able to describe new species for all these Scledoplatypus symbionts, and the Romances symbiont as well. So we were left with this situation here, and we wondered maybe this is the origin, right here in the red circle. Maybe this is where the domestication of ambrosia fungi in the Ceratocystadaceae first occurred. But there's actually a few problems with this. Three major problems that I'll outline. First are the non-ambrosial taxa that are interspersed between these ambrosial groups. Bretziella fagacearum is the oak wilt pathogen. The genus Huntiella are all wound-infecting saprophytes on wood. Ceratocystis adiposa causes black rot on sugarcane, and apparently at least once human disease, but I don't know about that. And Ceratocystis norvegica has an unknown biology. It was isolated from a bark beetle gallery, not an ambrosia beetle gallery, once in Norway, and it hasn't been seen since. So, kind of some weird groups, and there's no other known symbionts in here. These are kind of all by themselves. These are lone taxa with weird biology and surprising phylogenetic placement. But if we believe that these are placed correctly, phylogenetically, it would be hard to believe that these represent reversions from ambrosia symbiosis, going back to free living again. The ambrosia symbiosis is such a tight, obligate mutualism that once an ambrosia fungus has evolved towards dimorphic growth, mycangium yeast phase, food for beetles over the ability to even disperse on their own with spores, it would just be surprising if they were to revert to free living. And in fact, no ambrosia beetles are known to have reverted from ambrosia symbiosis to non-ambrosial symbiosis. Some of them have lost mycangia to steal fungi from others, but that's off topic. So the problem two is morphology. If we look across the ambrosia symbionts in the family, I've mentioned Illyrioconidia before. There are these distinctive globose conidia that are produced by some members. You see it in only some of the taxa. We certainly do not see them in Phyllophoropsis. They are not present in Huntiella. We did not see them in the African symbionts Wolfgangiella either. So again, we're seeing that morphological divergence in the family when, if it was a single ambrosial ancestor, maybe this would have carried forth, or they would have all been homogeneous. Finally, we decided to do a molecular divergence dating estimate to compare the divergence dates of these fungi to some known estimates of the beetles, specifically using the clades of beetles where the mycangium is known to exist as a representative of that mycangium's origin. So if we compare the origin of the pronotal disc, and I've got two different bars here to represent two different recent studies, one from 2017 and the other coming out this year. Both of those dates, both the solid and the dotted line, still are younger than the split of the ambrosia fungi in our analysis. So, with these three bits of evidence together, it was enough for us to reject the hypothesis that there was just a single origin in the family. And so we moved forward to seeing what the other possibilities might be. If I line up the other origins of the mycangium types here, you can see they actually line up pretty well with their individual symbionts. The mesonodal pouch mycangium origin just predates the origin of ambrosiella. The procoxal coil in blue just predates meredithiella in blue. And the prothoracic pleural just predates Phyllophoropsis in orange. So we thought maybe the origin was here at node 2 and node 3. After all, these both fall inside of the estimates, inside or younger than the estimates for the pronotal disc mycangium. So maybe symbiosis did start with the Scleroplatypodini, but twice, and was then passed on to these other lineages. So let's just focus on node 3 for now. Did Meredithiella, Toshinella, and Ambrosiella have a common ambrosial ancestor? The problem here is, or the most major problem, is that the genus Cortholis, the only one known to have that coiling tubular mycangium, are only found in the Americas, and they were never known in history to have been sympatric with the tribe Scleroplatypodini, which is in Asia and Africa. And when South America split from Africa about 100 million years ago, neither of these mycangia existed, and they don't appear to have had domesticated symbionts yet. So it would be really surprising to see some sort of common origin and dispersal of the fungi across the ocean. And of course, there's still the problem of the non-ambrosial taxa. Ceratocystis adiposa and Ceratocystis norvegica still fall inside of this origin. So I don't think this is very likely either. Instead, what if we had origins here? Meredithiella was its own separate origin onto itself over in the Americas, over in the New World, and Toshionella and Ambrosiella shared an origin with one another. Hey, the origin of Meredithiella lines up pretty well, right on actually, with the origin of its mycangium. And the origin here, the split between Toshionella and Ambrosiella, corresponds with both estimates for the origin of the pronotal disc mycangium. And if you look at the estimate for the mesonodal mycangium in green, it's younger, but it corresponds well enough with a symbiotic transfer, so a horizontal transfer from the Scletoplatapodini and their disc mycangia to these mesonodal pouch. And maybe this was the origin of the mesonodal pouch mycangia. Some ancient transfer of this already cultivated symbiont that allowed these beetles to develop these mesonodal pouches. This is interesting because if this is true, then this means not every mycangium origin necessarily corresponds with its own separate domestication of fungi, which was one of the hypotheses when we set out on this part. Moving back up to node 2 at the top, Phalapheropsis and Wolfgangiella. Was there an origin here at node 2? And what we're seeing now are the descendants of that single domestication. The problem here is that, as you can see, these are pretty distantly related groups. Their split is at about 30 to 35 million years ago, but the diversity within each of these genera is really small. This isn't that surprising because we had limited sampling of Scletoplatopus in Africa. Maybe we're missing a lot of diversity here in the tree. I wouldn't be surprised. But our sampling of the beetles with that prothoracic plural mycangium, the orange, was pretty comprehensive considering, both in taxa across the globe and in locations. Yet the origin of Phalapheropsis here is pretty young compared to the split between it and Wolfgangiella. And the mycangium origin here of the prothoracic plural is also still pretty young. So there could be unknown non-ambrosial intermediates here. One wouldn't be surprised if there's some unknown taxa out there. After all, we got a little lucky as it is in the rest of the family where we had these single lone non-ambrosial taxa by themselves. So there could be some intermediates hiding out there that would help us paint a better picture here at the top of the tree. Of course, it could also be that these represent a single origin and we're just sampling a small amount of that diversity, but instead, maybe these represent two separate domestications that were fairly recently. I can't say this confidently. We need more data. There certainly need to be more African Scletoplatopus collected and studied. It could also be that the diversity we need is extinct. And in that case, this is a question that might not be easy to answer. So I'm just going to put a big friendly question mark there. So in conclusion, the largest and most elaborate mycangia appear to have Ceratocystidaceae symbionts, either in or closely related to Ambrosiella. There is a surprising diversity of morphology here between the Allurioconidia of Ambrosiella and the deep-seated phyalids of Phalapheropsis. There were unexpected sexual states, the first reported in ambrosia fungi. And these symbionts are likely polyphyletic. They probably don't arise from a single origin of domestication by the beetles, but were instead domesticated at least 2, maybe 3 times by those beetles. There can be an interchange of fungus species among ambrosia beetles with the same mycangium type, and I want to harp on this for just a minute. All of the beetles with mesonodal mycangia had Ambrosiella. All of the beetles with prothoracic coiled mycangia had Meredithiella. All of the beetles with prothoracic plural mycangia had Phalapheropsis. But they don't seem to be species specific. So what we think we see here is a co-adaptation between fungal genus and mycangium type. We're starting with maybe a looser association, however many million years ago. They have co-adapted with one another over time to reach kind of a tuning with each other where you can trade the fungi between those types of mycangia but not with other mycangia. And finally, some beetle tribes may have acquired their symbionts horizontally from beetles that had different kinds of mycangia. I'm specifically talking about the pronodal disc mycangium of Scletoplatapodini and the mesonodal mycangium. So, moving back to my original analogy here of the Greek gods and their ambrosia, maybe some of them were being kind and sharing what they had with one another. Maybe a lot of the others were just kind of doing their own thing, if we're thinking of our beetles as Greek gods analogy. But here's the connection that I wanted to make. The Greek gods used their ambrosia for immortality, but if you look back at the history of Greek mythology, even Zeus only lasted a couple thousand years. And the ambrosia beetles have been using their ambrosia for going on 100 million years or so. So, I don't know, maybe that's real immortality. So, I want to say thanks, a genuine thanks to so many people not only Major Professor Thomas Harrington, my entire committee, all the wide range of collaborators across the world, without which any of this would have been possible. The IM and PLPM department and staff. Those of you who know me know I have been really active and involved with teaching, like Dr. Harrington mentioned. So everyone who has taught near me, above me, under me, in a proximity of me, thank you for that. Fellow students in MGSO, the Plant Pathology Graduate Student Organization, etc. Thank you so much, especially those of you that are here. And with that, I will take questions.